MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems, Passage 7, Initial Steps in the Metabolism of Dietary Monosaccharides. As you view the reading of this passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. Dietary carbohydrates are converted to three principal dietary monosaccharides: glucose, fructose, and galactose during their digestive process via the activities of enzymes that catalyze the hydrolysis of glycosidic linkages. This process takes place in the duodenum of the small intestine. Monosaccharides are then absorbed across the intestinal mucosa through transport proteins and are taken up into the bloodstream, where they travel to the liver via the hepatic portal vein and are transported into hepatocytes. Here, fructose and galactose are immediately phosphorylated through the activity of liver kinases, fructokinase and galactokinase, and are subsequently converted to glucose via the gluconeogenic metabolic pathway. Phosphorylated fructose can also feed directly into the glycolytic pathway. In contrast to fructose and galactose, hepatic glucose remains unphosphorylated for a longer period of time, despite the presence in hepatocytes of glucokinase and hexokinase. Unphosphorylated glucose is able to diffuse back out of hepatocytes and circulate throughout the body, where it serves as the principal substrate for cellular respiration in all tissues and cell types. Paragraph 2. The following graphs depict time course measurements of plasma fructose, glucose, and insulin after ingestion of 75 grams of glucose or 75 grams of fructose by human subjects. All right. Figure 1. Change in plasma glucose and fructose and plasma insulin in healthy human subjects following ingestion of 75 grams of glucose or 75 grams of fructose. The dashed line depicts change in plasma glucose after ingestion of 75 grams of glucose. The solid line depicts change in plasma glucose after ingestion of 75 grams of fructose. The dotted line, change in plasma fructose after injection of 75 grams of fructose. Okay? All right. Which of the following best describes the relationship between glucose and fructose? A. Enantiomers. B. Structural isomers. C. Epimers. Or D. Diastereomers. I'll give you a moment to think. Okay, so basically enantiomers are stereoisomers that have different configurations at all stereocenters. Okay, here goes our stereocenters. They are non-superimposable mirror images of each other, so if you were to fold this sheet over, they would overlap. but in and of themselves, they're non-superimposable mirror images. Diastereomers, those are basically stereoisomers that have different configurations at one or more stereocenters. Okay. But not all stereocenters, just one or more. And epimers are basically diastereomers that differ at one and only one stereocenter. So here we see that there's a flip in the configuration at carbon 2. Okay. All right. So basically, by default, the only answer choice left is answer choice B. That means that, let's see, glucose and fructose are going to be structural isomers. And structural isomers are molecules that share the same chemical formula but have different molecular structures. So glucose and fructose are structural isomers of one another. All right. Okay. Which of the following terms accurately describes the new functional group that results from the cyclization of the straight chain form of fructose? So what's this group? Is it A, a keto? Is it B, an acetyl? Is it C, a hemiacetal? Or is it D, a hemiketal? I'll give you a moment to think. All right. Okay, so ketones are functional groups consisting of a tetrahedral carbon with the connectivity C, R1, R2, OH, O attached to some R group, and the R group could be anything. Okay? R denotes like anything. Acetals, however, are functional groups consisting of a tetrahedral carbon at the center with the connectivity C, H, R, OR, OR. These R's could be anything. They're derived from aldehyde groups. I'm sorry. That's hence the name acetal. Okay. Hemiacetals have the connectivity C, R, H, OH, OR. Okay. Hemi because this R is an H. And hemiketals have, these are functional groups, and they have the connectivity consisting of a tetrahedral carbon again, attached to two different R groups, as well as an OH, hence the name hemi, and OR. Okay, hemi meaning this H attached, the R group attached to one of the O's is an H, a hydrogen. So you have some type of alcohol, okay? So the cyclical form of fructose, fructofuranose, is basically a hemiketal functional group. All right? Which of the following choices comparing the Km and K catalysis for liver fructokinase and glucokinase. Remember, kinases are enzymes that phosphorylate molecules, would definitely result in the properties described in the passage, assuming both enzymes have similar concentrations and Vmax. A, fructokinase has a higher Km and a lower K catalysis. B, fructokinase has a lower Km and a higher K catalysis. C, fructokinase has a higher Km and a higher K catalysis. Or D, fructokinase has a lower Km and a lower K catalysis. I'll give you a moment to think. All right. Okay, so if you're not familiar with reaction kinetics, this question can seem a little daunting. However, it's really not that bad. So let's first start off with defining each of these variables. The Michaelis-Menten constant, K sub m, of an enzyme is defined as the substrate concentration at which the conversion from reactant to product is half the maximum rate. Okay, so hold that thought. And intuitively, the lower the Km of an enzyme, the faster it converts reactants to products at non-saturating substrate concentrations, okay? Now, K sub cat, which is short for K catalysis, of an enzyme is the number of molecules of substrate converted to product per molecule of enzyme. So how much juice can you get out of an enzyme per second? Okay, so how much do you get out of it per second? So basically how fast is your enzyme working? Then Vmax is defined as K catalysis times enzyme concentration. Okay, so it's going to tell you the maximum speed at which it's like fully loaded. Okay, so you're probably wondering, okay, how the heck am I going to figure this out? Well, there are actually a few key clues and keywords mentioned in the passage. You don't have to look at the graph for this one. It's actually spoken in paragraph 1. Okay, so the second half of paragraph 1 starts out, well, it doesn't start out, it's like it mentions that hepatocytes, okay, so here in hepatocytes, fructose and galactose are immediately phosphorylated. And we know that fructokinase is the phosphorylating enzyme for fructose. So with that thought alone, we know that it's going to have a lower Km. Okay, we're just considering Km, not K catalysis yet. So we can eliminate answer choice A and answer choice B. Okay, so assuming that both concentrations of glucokinase and fructokinase are similar, we already know that fructokinase, well, we're assuming that fructokinase has a lower Km, because if something works immediately, that means you don't need a high concentration to get it going, okay? So I'll continue reading. Here, fructose and galactose are immediately phosphorylated through the activity of liver kinases, fructokinase and galactokinase, but we're not concerned with galactokinase, we're concerned with glucokinase, so we have to keep on reading to see where glucokinase is mentioned, and are subsequently converted to glucose via the gluconeogenic metabolic pathway. Phosphorylated fructose can also feed directly into the glycolytic pathway In contrast to fructose and galactose, hepatic glucose remains unphosphorylated. Okay, so this confirms our thoughts that fructokinase has a higher Km than glucokinase, because if glucose is in the presence of glucokinase, but it's remaining unphosphorylated, that means that you need basically a high concentration of glucose to get things going for glucokinase. Okay. For a longer period of time, despite the presence in hepatocytes of glucokinase and hexokinase, unphosphorylated glucose is able to diffuse back out, blah, blah, blah. So from this, we can also make some inferences about K-catalysis between the two enzymes. So we can assume that the K-cat for fructokinase is going to be higher than the K-cat for glucokinase. Okay, so yeah, the correct answer choice is answer choice B. Grams of sucrose is administered instead of glucose or fructose. Which of the following is the most likely outcome for circulating glucose, fructose, and insulin? Is it A, circulating levels of glucose and fructose would increase to similar levels as those depicted in Figure 1? Circulating levels of insulin would increase to higher levels than those depicted in Figure 1? Is it B, circulating levels of fructose, glucose, and insulin would increase over time, but only to levels about half as much as depicted in Figure 1? Or is it C, circulating levels of each of the three molecules would not increase? I'll give you a moment to think. All right. Okay, so sucrose is a disaccharide consisting of one molecule of glucose and one molecule of fructose, joined via a glycosidic bond, as shown here. Administration of 75 grams of sucrose amounts to administration of 37.5 grams of glucose and 37 grams of fructose. So circulating levels of fructose, glucose, and insulin would increase over time, but only to levels about half as much as those depicted in Figure 1, okay? So this, might seem a little daunting at first, but it's pretty straightforward, all right? What changes to Figure 1 would one expect to see in a patient suffering hepatic fructokinase deficiency in which the fructokinase enzyme in the liver is inactivated? Okay. A, circulating fructose would more closely mirror the rise of circulating glucose, but would fall more rapidly. B, circulating fructose would more closely mirror the rise and fall of circulating glucose. Or C, circulating fructose levels would more closely mirror the rise of circulating glucose levels, but would remain high, unlike glucose levels. I'll give you a moment to think. All right. Okay, so let's reason our way through this. Phosphorylation of fructose serves to trap the fructose that has diffused into the hepatocytes. It also maintains a concentration gradient for fructose that favors the continued diffusion of fructose into hepatocytes. For those of you who don't recall, hepatocytes are liver cells. Failure to phosphorylate fructose in hepatocytes results in high levels of circulating fructose, okay? It is also necessary to phosphorylate fructose before it can enter into any metabolic pathways such as glycolysis or gluconeogenesis. So circulating levels of glucose, they're going to, in this case, they're not going to remain high because insulin drastically increases the capacity for glucose to diffuse into cells where it can be phosphorylated and thus enter into metabolic pathways. So overall, the answer is going to be circulating fructose would more closely mirror the rise of circulating glucose, but it would remain high because, it's just going to continue circulating. All right? Okay.